Hi, my name is Jacob Schwartz, and today I'm presenting on behalf of myself, Eric Bell, and Peter Fredolino, our poster for ISMB, two approaches for predicting dimer protein structure from multiple sequence alignments using deep residual neural networks. The central idea behind these projects is that while AlphaFold and RosettaFold have essentially solved the single-chain protein structure prediction problem in the past 2 years or so, the problem of higher order protein complexes, predicting their structures, is not quite solved. And since proteins rarely act independently and often form complexes to carry out biological functions, this problem is very important. When I speak of two approaches in our title, I'm referring to two separate pipelines for homodimer structure prediction and for heterodimer structure prediction. These are two separate approaches because these are essentially two different problems, as I'll later describe. So first, in the case of homodimers, it's been shown in previous work that when a monomeric MSA is made for just one chain, if it forms a homodimer, the homodimer contacts have their potentials hidden within the coevolutionary information of the monomeric MSA. Theoretically, if we had the monomeric structure solved and we had a pipeline like deep potential which predicts the monomer's residue-residue contacts, we could figure out which predicted residue-residue contacts refer to dimerization rather than an intrachain contact. This pipeline seeks to extract the interchain contacts from multiple pipelines' monomer structure predictions. Specifically, we use AlphaFold to create a structure that is a proxy for a solved structure. We use deep potential to be the main predictor from which we'd like to subtract out the intrachain contacts. And we additionally introduced the MSA transformer, which was out of Facebook's machine learning research lab, to introduce some more diversity into our data. Our input ends up being 3 different distance maps from 3 very different sources. And we expect that these data will be wrong in different ways, and that our residual convolutional neural network should be able to extract the true interchain contacts from the differences in these 3 inputs. So once the monomeric MSA goes through each of the structure prediction pipelines, in the case of deep potential, we directly get a distance map and some angle maps. In the case of AlphaFold, we get a structure which we will convert back into distance and angle maps. And the MSA transformer ISMB will also give us distance and angle maps. We stack the three-dimensional data, and we put that through many residual blocks, each of which consists of a convolution and normalization step followed by an activating function, get another convolution and normalization step, and then a residual step with another activating function. This should generate distance and angle maps from which we can solve the structure given monomeric structure predictions as well. Now moving on to the heterodimer pipeline, as I mentioned before, this is a fundamentally different problem. Since the 2 chains are not identical and we need to find the information between them, this requires construction of a paired MSA. It has become somewhat of a standard throughout this field to use species-based pairing, which is moderately naive in its assumptions. Specifically, species-based pairing assumes that 2 protein sequences, which have the same species origin, co-localize in that organism. We know this is not true in all cases, and it can introduce some noise into the data to pair proteins in the same species, which are not ever in contact in that species. Also, we now have access to very large sequence databases, specifically from metagenomics, where we don't know the species origin of these sequences. so species-based pairing makes usage of these large data sets impossible. Through our work, we hope to capture the sequences of the metagenomic data sets into our training to achieve higher accuracy. We also hope to be able to work with species-agnostic pairings so that we can capture interactions which happen interspecies. So first, we have classifier-based pairing. We hope to use a conjoint triad classifier, and from that we take the sequence pairs which have the highest likelihood of interaction according to the classifier and include that in our pairing. The second way we hope to pair is through coevolution maximization. And through that, we use separate heuristics to algorithmically maximize covariance within an MSA. We hope that this will capture all possible coevolution which can happen between sequences, although we recognize that such algorithms will take very long to compute and might introduce false pairings which will be noise in the data. To account for the possible faults in these three pairing methods, we hope to build a consensus which will use these three methods to put together the best possible paired MSA, which will work better than the current state of the art. Once we have a paired MSA, this pipeline is an expanded form of the deep potential pipeline out of the Zhang lab at the University of Michigan. Deep potential is the successor to res triplet, triplet res, and res pre, and without this paired step simply takes a one-dimensional ResNet from a monomer and combines that with the mutual information and pseudo-likelihood maximization matrices in a two-dimensional ResNet to predict intrachain contacts. This pipeline splits into a paired MSA part and two monomer parts. So those two monomer parts are one-dimensional ResNets, which process the HMM, the self-mutual information of the individual MSAs, and the PLM field parameters, each through 10 residual blocks, and then tiles them to be included in the larger 2D model. Through pairing, these MSAs are then put through CCM-pred and a direct mutual information calculation function to create this large three-dimensional input to the network, which is independently put through 10 residual convolutional blocks. We combine the outputs from these two trunks of the pipeline into this part here, our two-dimensional ResNet, and put that through 40 further residual convolutional blocks. This very large network results in a set of different predictions of residue-residue distances between chains, as well as dihedral angles between the chains. Given these maps, in conjunction with monomer structure predictions, we will be able to construct the heterodimer structures of these proteins. In conclusion, these are two separate novel approaches for predicting dimer structure from sequence. They each rely on multiple sequence alignments and deep residual neural networks, but work in fundamentally different ways because homodimer structure prediction and heterodimer structure prediction are fundamentally different problems. Thanks so much for listening to my presentation today, and I hope to see you at ISMB.